Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Today we are delighted to welcome Dr. David Belin from the Department of Psychology here at Cambridge University. David is a reader in behavioural neuroscience and a fellow and director of studies at Homerton College. Today, David will speak to us about his research on social deprivation, coping and drug addiction, a bad cocktail in the COVID-19 era, sharing evidence from preclinical studies with us. Thank you, David.
Thank you, Davila. It's a great pleasure to be here, and I thank you all for attending this talk. Once again, I apologize for the technical glitch that forced me to postpone the talk to today. So we're going to discuss together how we've implemented procedures in animals to try causally to determine the extent to which experiencing drugs in a context of social deprivation or negative affective states that lead to the use of drugs as a coping mechanism does indeed facilitate the transition to addiction. So first of all, what's drug addiction? So you may have all heard about the fact that all addictive drugs increase dopamine concentration in the ventral striatum or the linx accumbens, and it has long been suggested that the addictive properties of all these drugs were due to their ability to trigger feelings of pleasure and euphoria in the individuals who take these drugs. However, some individuals do take these drugs for other reasons, such as the use of alcohol as an anxiolytic strategy or a disinhibition strategy for individuals who feel the need to medicate these particular states. But what is important to bear in mind is that addiction is not just about taking or seeking drugs. Many of us do take drugs. I do have coffee here. There is whiskey in my office, but I'm not drinking it right now. And despite several years of drinking alcohol, I want to think that I am not yet addicted to alcohol, because addiction is characterized by the compulsive pursuit and consumption of drugs. Individuals with an addiction would actually harm themselves, lose their family, lose their job, lose their life due to drug use. And it is very important to bear in mind that we are not equally vulnerable to addiction. As a matter of fact, of 100 potential users, only some will be initially already vulnerable to developing drug addiction. Of these users, some would try specific drugs and like them, and they may move on to recreational use. Some already do not necessarily like these drugs and would not pursue further the use of these drugs. Of those who use drugs recreationally, only a few would eventually abuse them, and only these individuals who were already vulnerable prior to drug use would eventually switch to addiction. The issue is, once they've established an addiction, they have a very high risk of relapsing to taking the drug, even after prolonged periods of abstinence. So drug addiction is a compulsive relapsing disorder, and it's very difficult to relinquish the use of drug once you've developed an addiction. So this inter-individual variability in the vulnerability to developing drug addiction suggests that there must be something in the brain that renders specific individual prone to developing drug addiction. And clearly, addiction has to be considered as the interaction between a set of genes, their interaction with a specific environment, and that contributes to shaping specific personality traits, and eventually the interaction with the drug. And it is the interaction between genes, the environment, and the drug that is going to shape this individual tendency to lose control over drug use and develop drug addiction. But the issue is that it's a burning question for me. It's been what the question with which I started doing research. What are these factors that convey this increased vulnerability to developing drug addiction? And actually, with the COVID-19 situation, you can't read a magazine or newspapers without finding a lot of articles about the extent to which loneliness and isolation fuel alcohol addiction, even in older people. The fact that the use of drugs is promoting addiction under COVID-19 related lockdown has been coined disease of despair. And in the UK particularly, problem drinking has been soaring under UK lockdown. And that strongly suggests that the loss of social interactions and the stress related to self-isolation has been promoting further drug use and very likely exacerbating the transition to drug addiction. As a matter of fact, not only in magazines, but in real journals as well, in scientific journals, such as this one, you can see that it's been now clearly established that people with substance use disorders are at greater risk of worse COVID-19 outcome. And there's been a surge in addictive behaviors, both new and relapse to usage of drugs, including behavioral addiction during the lockdown periods. Withdrawal emergencies and death are also being increasingly reported during that period. So there is really there an important societal consequence to the response to that pandemic and a major impact on the mental health of many individuals who are at risk of developing a drug addiction. And it is quite important to understand the extent to which indeed it is the influence of social deprivation and the motivations to utilize drugs as a means of coping while individuals experience lockdown or the related stress that exacerbates such vulnerability to developing drug addiction. And of course, it is very difficult to control these various factors in human studies. So one can rely on the interest of longitudinal studies in animals. So what we will see today together is whether we can causally establish that the use of drugs under a state of deprivation of exteroceptive sensory stimulation and social isolation indeed facilitates the transition from controlled to compulsive drug use. We'll see whether we can causally demonstrate that the reliance on drugs to cope with negative affective states, such as those experienced under COVID-19 related conditions, exacerbates addiction vulnerability. And clearly, in order to answer these questions, we must be able to measure individual vulnerability to develop compulsive drug intake and parametrically control and manipulate the environment. And I'll try to share with you how we've managed to implement these particular experimental conditions in the lab. So there is great interest in using preclinical studies to address such questions, because when you think about it, every one of us came to Earth with a set of genes that were already shaped by the experience of our parents through epigenetic adaptations. And these genes have been interacting with an environment throughout the neurodevelopmental period to shape specific personalities and coping strategies. But it is only once the individuals have developed a pathology, in this particular case, drug addiction, that they are identified once they are actually enrolled into the NHS and medical care system. And it is not possible retrospectively to understand how these individuals were prior to the development of this aberrant behavior. So what we've attempted to do was to translate the human situation into a rodent situation, especially in rats. Because rats, they also come to Earth with a set of genes, and we can control their environment. And I will try to show you that rats do have specific behavioral features that are reminiscent of personality in humans. And when exposed to drugs, only some of them, not all of them, are going to develop maladaptive behaviors that share construct validity with the diagnostic criteria of drug addiction as defined in humans. The question is, how do you measure addiction in rats? For many years, the gold standard was the so-called self-administration procedure. In this procedure, a rat that is equipped with an intrajugular, intravenous catheter that protrudes from its back. This catheter is connected to a tubing, itself protected by the wire here, and the tubing is connected to an infusion pump here and a syringe. And in that syringe, there's a drug such as cocaine or heroin. And the animal is going to work. In this video, you'll see the rat pressing on the lever here, and he's just here received an infusion paired with this stimulus in the environment called a conditioned stimulus that is associated with the delivery of the drug. And that has for a long time been the gold standard for preclinical research for drug addiction. But actually, what that is the model of is a rat taking drugs in a cage. That falls very short of recapitulating the maladaptive nature of drug seeking and taking behavior that is characteristic of addiction in humans. So during my PhD, what I tried to do was to take the clinical definition of drug addiction as per the DSM, the Diagnostic and Statistical Manual of Mental Disorders. And at the time I did my PhD, a long time ago now, it was the DSM-4, now it's the 5th version, but nevertheless, the various criteria in the DSM were tolerance and withdrawal, and then 5 more criteria, which together reflect the fact that individuals are not able to maintain control of their drug use. They would show a persistent desire or unsuccessful attempts to cut down drug use. They would show a great deal of time spent obtaining drugs or recovering from its effects. They would give up other activities because of drug use. And importantly, they would continue drug use despite awareness of harmful consequences. And we have established behavioral procedures that enable the operationalization of these criteria in rats. We could measure the inappropriate persistence of drug seeking in rats, even if the drug is not available and the rats know it is not available. We could measure the aberrant motivation shown by these rats towards the drug by asking them how much they would be willing to pay for a single infusion of the drug. And these procedures are called progressive ratio schedules of reinforcement, and we play a little bit with their behavioral flexibility, their behavioral economics. So every day the rats are trained such that each drug infusion costs them 5 liver presses. And on that particular day, the first infusion cost them 5 liver presses, but the second one cost them 20. And the third one cost them 80. And at one point, the rat is going to give up. And that break point is a good index of its motivation, how much he is willing to pay for a single infusion of the drug. And to determine the extent to which animals would persist in taking drugs despite negative consequences, we measured their tendency to continue responding despite food shock mediated punishment. Okay? And by combining these 3 criteria, we could identify within large cohorts of animals rats that are positive for each one of these criteria. I remind you that in humans, you need to meet at least 3 criteria of the 7 out of the previous 12-month period. So here we had 3 criteria. A rat that actually fulfills or matches the 3 criteria is called the 3 criteria rat. And of course, it will be deemed addicted to the drug. In contrast, some rats would never be positive for this criteria, and they will be called 0 criteria rats. As you can see here, there is a linear relationship between the number of criteria these animals show and the addiction score. Importantly, the 3 criteria rats here, the addicted rats, had as a group an addiction score that went beyond the standard deviation of the population. So as a group, they really drifted away from, let's say, normative or normal behavior. And this is what it looks like for their motivation, their perseverance of drug seeking, and their resistance to punishment or compulsivity. There was a criterion-dependent increase in behavior. And indeed, the 3 criteria rats displayed much higher behaviors in each one of these 3 criteria as compared to 0 criteria rats. When we first identified this phenotype, it was surprising to us that the 3 criteria population represented between 15 and 20% of a large cohort of animals. And that incidence of aberrant drug self-administration behavior was very similar to that observed in ongoing drug users in the case of cocaine. Having established this phenotype, it was quite interesting to see that the addicted rats and the non-addicted rats, prior to being challenged for these specific addiction-like behaviors, did not differ in the total amount of drug they've taken. So these individual differences were very much the result of the interaction between predetermining factors and the same exposure to the drug. And we further established that if you gave these rats extended access to the drug for 5 hours nonstop, the 3 criteria rats would escalate their drug intake much more than the non-addicted rats. And that is a behavioral manifestation of the inability to control intake, loss of control over intake, which was another criterion for drug addiction. So that evidence supported the idea that identifying rats on these 3 behavioral manifestations was also predictive of an additional criterion of drug addiction, such as loss of control. To further establish the predictive validity of that procedure, following a period of abstinence, we tested the propensity these rats showed to relapse to drug self-administration, and we challenged them with increasing doses of cocaine delivered non-contingently, and the extent to which, in response to these increasing doses of cocaine infusions, they would seek the drug, they would respond for the drug. And you can see that the addicted rats respond much more than the non-addicted rats. So overall, this model has good construct and predictive validity with regards to the individual vulnerability to developing drug addiction. So now we have that model that enables the identification of specific vulnerability to develop the pathology. We were in a position to carry out longitudinal studies in which we could test factors of vulnerability to developing that specific behavior. And we started with something that you may all be familiar with, the sensation-seeking personality trait. It's commonly considered that sensation seekers are crackheads. But as a matter of fact, if you look at the sensation-seeking scale, there are 2 specific subdimensions to it. Boredom susceptibility, which is I get restless when I spend too much time alone. That speaks a lot to the COVID-19 situation, right? I prefer friends who are excitingly unpredictable. And thrill novelty seeking. I would like to try bungee jumping, or I would like to do frightening things. Importantly, boredom susceptibility or boredom proneness is only marginally related to sensation seeking. But in studies that have aimed at establishing its relationship with other emotion regulation or emotion-related processes, it has been established that boredom proneness correlates with anxiety, depression, hostility, and correlates most with dysphoria. Okay? So boredom susceptibility may well represent an additional gateway towards developing drug addiction if individuals were to use drugs to medicate this dysphoric state. Importantly, what has been shown in a very elegant study is if you were to present humans, participants, with a borrowing film fragment as opposed to a neutral film fragment, before the borrowing film fragment exposure, they would report boredom, fatigue, and frustration at random, actually baseline levels. And exposure to the borrowing film fragments would substantially increase their boredom report, their fatigue, and their frustration. Statistically so as compared to a neutral film fragment. What was shown by Vodanovic and Werner is that individuals experiencing this exacerbated boredom would eat much more M&Ms as compared to presentation of a neutral film. That is a food-related coping strategy. Importantly, if they were given the opportunity to self-administer electric shocks to deal with that boredom, individuals would self-administer shocks following the presentation of the boring film as compared to the neutral film. That demonstrates the extent to which boredom can be a very powerful drive towards negative reinforcement. Individuals would even self-administer shocks to avoid boredom. So boredom susceptibility and novelty seeking can be measured in animals, in two different tasks. The first one is to expose rats to a new environment, exciting environment. And these are here 4 open fields. These are 50 centimeter squares. And you can see in red the cumulative trajectories of these rats. Some rats are not going to explore the environment much, and others, they are going to respond a lot to that environment. They are very excited and they explore it a lot. Those who explore the environment a lot are called high responders, and those that explore the environment less are called low responders. And that locomotor reactivity to novelty has been shown to predict in a rat an increased tendency to take drugs. In contrast, you can measure boredom susceptibility by offering a rat in a 3-compartment box like this, a choice between a known boring compartment and a new exciting compartment. So you would place the rat for 30 minutes in one compartment. Immediately after that, you would place the rat in the central corridor, and you give the rat free access to either compartment. And you would measure the extent to which the rat will spend time in the new compartment, or in other way, avoid time in the boring compartment. And there are some rats that are called high novelty preference rats, highly sensitive to boredom, and rats that would spend much less time in the new compartment. There are low novelty preference rats. So in the next experiment, what we did was to characterize rats based on their sensation seeking or locomotor reactivity trait or their boredom susceptibility trait as high responders, low responders, low novelty preference, or high novelty preference. Following 100 days of drug self-administration, we tested them for their addiction-like behavior. And as you can see here, high responders and low responders, so the thrill-seeking traits, there were no differences between them with regards to addiction, and their addiction scores were, if anything, negative. In contrast, high novelty prefer rats, so these rats who were sensitive to boredom, showed highly positive addiction scores. These addiction scores being the combination of the 3 specific behavioral features I've referred to earlier on. We then looked at which one of these behaviors predicted best this addiction score in these high novelty preference rats. And as a matter of fact, high novelty preference rats showed higher motivation, higher persistence of drug seeking, and higher compulsivity than low novelty preference rats. And there were no differences whatsoever in any of these behavioral traits between high and low responder rats. So that is evidence that high novelty preference in the rat is a factor of vulnerability to switch to compulsive drug self-administration, and in that case, cocaine self-administration. And that is in marked contrast with another evidence from the lab from the time I was Barry Everett's postdoc. We tested the extent to which high responder rats, so sensation seekers, as opposed to low responder rats, are indeed differentially prone to acquire drug use. And we challenged these animals to acquire drug self-administration with increasing doses of cocaine. And you can see in red here that high responder rats, those that are thrill seekers, would self-administer a large quantity of cocaine at doses which do not actually support responding more than that of saline in low responder rats. So what we have here, we have one behavioral trait, thrill-seeking or high locomotor response to novelty, that predicts an increased tendency to use drugs, to engage in drug use, and another trait, boredom susceptibility, that instead predicts an increased vulnerability to switch from controlled to compulsive drug use. And that is evidence that the factors that predict an increased tendency to acquire drug use are dissociable from those conferring an increased vulnerability to addiction. And to the best of my knowledge, it was one of the very first suggestions that these factors were absolutely dissociable. And it also suggests that situations that promote boredom, considering the role of boredom in the vulnerability to addiction, boredom can be identified or defined as the lack of exteroceptive sensory stimulation, may facilitate the development of addiction through maladaptive coping. Okay? So now we've established that there were already factors of vulnerability. It was time to determine the role of the environment and the role of social deprivation on these particular factors. So this is when we delve into the COVID-related question and the extent to which COVID-related lockdown or deprivation of exteroceptive and social stimulation triggers coping mechanisms that exacerbate the vulnerability to addiction. And I'm going to show you two sets of experiments. The first one was designed to address that question. Does the deprivation of social exteroceptive sensory stimulation facilitate the transition from controlled drug use to compulsive drug use? And the second one is, does the reliance on drugs to cope with distress experienced in a drug-taking context exacerbate the vulnerability to develop drug addiction? In the first case, we will artificially trigger contrasts between housing conditions, having rats in an enriched environment with a lot of social interactions, a very fun and exciting environment with a lot of exteroceptive input, as opposed to rats housed in pairs in standard laboratory cages. And having these differential housing conditions, we could contrast the extent to which they experienced drugs in a drug setting, which was common between these two, but animals would go from here to there to experience the drug, or from such an environment to there to experience the drug. And we had introduced like this, using the environmental enrichment as an increased contrast between housing conditions and drug setting. The second experiment I will report to you is one that was carried out by Luthia Martiprat's in lab. This has been a major endeavor, and I'll refer to the various people involved. Luthia here developed a new procedure in which she measured the acquisition of alcohol use as an adjunctive response, as a coping behavior. And she measured the extent to which animals that relied on alcohol to cope with distress would be more vulnerable to developing compulsive alcohol drinking than other animals whose means of coping were more adaptive. So first experiment, gene, drugs, environment interactions in the context of cocaine addiction. So in that experiment, we took 48 rats, a large cohort of animals, half of which would be housed in the standard environment, and the other half in the enriched environment. And what was done by Maxime Poussac, Nathalie Van Heel, Michael Puot, and Eric Ducré initially was to test the effect of these differential housing conditions on several behavioral traits that are relevant to the vulnerability to drug addiction. The first one is saccharin preference or sweetness preference. It's a proxy for determining the hedonic state of these animals. Then we measured their anxiety in the elevated plus maze. I'm more than happy to answer questions about these specific tests afterwards. We measured their thrill seeking, their locomotor activity to novelty. We measured in an auto-shaping task the extent to which these animals are prone to ascribe incentive motivational salience to environmental cues. We know that in drug addiction, drug paired cues are capable of driving the behavior, and there are inter-individual differences in the way organisms ascribe incentive salience to these cues. So we tested that particular trait. We then tested their boredom susceptibility in the novelty preference task, and we gave them access to cocaine in a cocaine self-administration regimen. What we observed is that housing conditions per se, a rich social life with a lot of exteroceptive inputs, changes completely the multidimensional behavioral profile of these animals. I would call it a pseudo personality. And this is what you can see here on this polar chart. Each one of these axes represents a specific trait: thrill-seeking or locomotor activity, anxiety, sign tracking, this auto-shaping procedure, novelty preference, and sweetness preference. in a standard environment, you can see that thrill-seeking is a prominent determinant, a prominent characteristic of the behavioral profile of the animals. And in the enriched environment, that trait completely disappears. Relatively speaking, it is now sweetness preference and novelty preference, which in the enriched environment personality model drive most of the variance, whereas it was locomotor activity to novelty and anxiety that were driving the variance in the standard environment. So clearly, housing conditions and the extent to which individuals are exposed to social interactions and exteroceptive stimulation changes the behavioral profile or the pseudo personality of these rats. And I would like to focus your attention on the fact that the enriched environment did not influence much novelty preference, so boredom susceptibility, but it did substantially decrease the trait of thrill-seeking or locomotor activity to novelty. And if you remember, locomotor activity to novelty is a trait that predicts an increased tendency to take drugs, but resilience to drug addiction. And indeed, we demonstrated, as previously shown, that rats raised in an enriched environment are much less likely to acquire drug self-administration, cocaine self-administration, as opposed to rats raised in a standard environment. This is partially due to the decrease in the locomotor activity to novelty trait, as well as an effect of the enriched environment on the low novelty preference trait. Low novelty preference rats that are otherwise also resilient to addiction were shown to be highly sensitive to the enriched environment and self-administer much less drug than high novelty preference rats. Those high novelty preference rats raised in enriched environments did not differ from low or high novelty preference rats raised in a standard environment. So that first experiment enabled us to demonstrate that the housing conditions per se shape personality or pseudo-personality in rats and promote an increased resilience to taking drugs in animals that would be more prone to take drugs but resilient to addiction. In a second experiment, we then tested the hypothesis that the environmental contrast between the housing conditions and the drug setting context would influence the transition to addiction. So in that experiment carried out by Maxime Poussac, Michael Pou, and Eric Ducret, we set up a new procedure to do self-administration in animals housed in groups. And these rats were housed either in an enriched environment or a standard environment. They received implantation of catheters, and they were trained for more than 50 days to self-administer cocaine. At the end of this long exposure to cocaine period, we measured their behavioral traits of the behavioral features of addictions. And we could identify from that cohort rats that showed 3 criteria, they were addicted to cocaine, and rats that showed none of these criteria, they were resistant to cocaine. Once again, the 3 criteria group had an addiction score that was beyond and above the standard deviation of the population. Once again, these different groups did not differ in terms of total intake of cocaine prior to the characterization of their compulsive behavior. Very interesting outcome of that experiment. What we characterized when we retrospectively looked at which environment came these various rats from, rats from the enriched environment showed positive addiction score overall, whereas rats from the standard environment showed negative addiction score, thereby suggesting that the relative enrichment of the housing conditions and the increased contrast there was between the housing conditions and the drug taking setting, or in other words, the extent to which animals were experiencing the drug in an impoverished social and exteroceptive environments, drove an increased vulnerability to addiction. And when we then looked at the representativity of animals in the enriched environments as compared to the standard environments, in the addicted population, the 3 criteria population, all the rats that had developed addiction came from the enriched environment. whereas the majority of the rats that were resilient to addiction came from the standard environment. And this differential vulnerability to addiction was due to an effect of the enriched environment on compulsivity, persistence of drug seeking, and motivation. So each one of the three behavioral features of addiction were influenced by these differential housing conditions and the consecutive increase in contrast between the housing conditions and the drug taking context. These differences were not due to a difference in cocaine intake between the two groups, and they were not due either to an influence of housing conditions on pain sensitivity. I remind you that we measure compulsivity as the tendency or the propensity to persist in responding despite despite punishment by electric shocks. So we controlled that the differential housing conditions had no effect on pain sensitivity. So together, these data demonstrate that exposure to cocaine in a state of loss of exteroceptive sensory stimulation or rich social interactions results in an increased vulnerability to switch to addiction. We artificially enriched the exteroceptive nature of the housing conditions and the richness of the social interactions within the housing conditions in relation to the drug-taking context using an enriched environment, and that resulted in an increased vulnerability to switch to addiction. So now the question is, does the deprivation of the exteroceptive sensory stimulation or social interaction also exacerbate the vulnerability to develop compulsive alcohol drinking, whether it exacerbates the vulnerability to addiction to another kind of drug, such as alcohol? And of course, stimulant drugs such as cocaine and alcohol do not at all have the same brain mechanisms, and alcohol is much more used as an anxiolytic coping strategy than cocaine. How do we measure compulsive alcohol drinking in rats? So here, Lucia carried out another experiment in rats trained in what is called a two-bottle choice experiment. They had access to a bottle of alcohol or a bottle of water. And they had access to alcohol or water. They had access to water all the time, but to alcohol or water every other day. So this is called intermittent access to alcohol. And you can see on this chart here, over daily sessions, the alcohol intake of these rats goes from 2 milligrams per kilogram in 24 hours to 6 milligrams per kilogram in 24 hours. They do escalate their alcohol intake. But that, again, falls very short of recapitulating the criteria for drug addiction. So we tested whether these animals would persist in drinking alcohol despite negative consequences. In that case, we rendered alcohol bitter with quinine. So we made the alcohol solution taste much less good. And that is called adulteration with quinine. And we also determined whether these animals would show a narrowing of interest for other sources of reinforcement. And we measured the extent to which they would persist in drinking alcohol despite an alternative reward. Okay? We carried out a cluster analysis, which to some extent structures a scatter plot like this into specific subpopulations. And you could identify animals that were sensitive to adulteration with quinine and sensitive to the introduction of an alternative reward. So, they would stop drinking alcohol if something else is offered, and if it doesn't taste good, they would completely stop drinking it. And there are animals here that would resist to either the adulteration of alcohol or the opportunity to drink a highly rewarding alternative reinforcer such as saccharin. So they were sensitive animals or resistant animals. And of course, animals that were resistant to adulteration and saccharin persisted in responding and drinking alcohol, whereas animals that were sensitive decreased substantially their alcohol intake in both tests. So the animals that were resistant to both these challenges displayed behavioral manifestations that were reminiscent of the compulsive nature of alcohol drinking in humans. Importantly, what was shown by Lucia is that the compulsive rats were characterized by an alteration of the enzyme that is responsible for the homeostasis of GABA in the central amygdala. And the central amygdala is part of that system that is involved in the fight or flight response, but also mediates a lot of stress-related mechanisms in the limbic system. So these animals could be characterized at the neural systems level by a specific alteration of one of the brain structures involved in regulating emotional responses to stress. Having established that procedure, we tested the hypothesis that the environment facilitates the transition to compulsivity. So here, Maxime Froussac and Solène Hansker had rats raised in an enriched environment or standard environment, and they were given intermittent access to alcohol. And we measured, after different forms of differential access to alcohol, their propensity to relapse following an abstinence period of 20 days. And again, here, animals in the enriched environment showed an increased propensity to relapse as compared to animals in the standard environment. So having increased the contrast between the housing conditions and the drug-taking conditions, we promoted an exacerbation of the vulnerability to develop compulsivity, to develop relapse. And when these animals were challenged at relapse to drink alcohol despite adulteration with quinine, animals in the enriched environment were much less sensitive to quinine adulteration, which is the cue here, than animals in the standard environment. So yes, increasing the contrast between housing conditions and drug-taking contexts, therefore, increasing the extent to which animals were deprived from social stimulation and exteroceptive stimulation when engaging with drinking alcohol, promoted an exacerbation of drug addiction. So exposure to cocaine or alcohol in a state of loss of exteroceptive sensory stimulation or rich social interactions results in an increased vulnerability to switch to addiction. So does it mean that the reliance on drugs to cope with distress, the distress experienced in the face of loss of social interactions in a drug-taking context, results in a facilitation of the development of addiction? Is it true that we can causally establish that alcohol use as a coping mechanism facilitates the transition to addiction? Here, we used another procedure, which is called schedule-induced polydipsia. Those of you who like sweet things had this talk being given outside the COVID situation. I want to believe that after the talk, if you were not too tired of my French accent, we would have been for a drink and perhaps to the restaurant. What about this situation? You go to the restaurant and we're just waiting for this nice dessert we've been dreaming of since the beginning of the meal. And you're just about to have your dessert and say, no. I'm going to give you half a teaspoon of that dessert every 2 minutes. Would you enjoy it? Or would you experience this burst of frustration and you would like to slap my face, take the spoon and eat your dessert your way? Well, to some extent, this is what we do to these rats. These are food-deprived rats that are brought to a cage and they learn that they can have as much food as they want. In that cage, there is a bottle of water readily available here. And one day we take control away from them. We deliver 45 milligrams of food every minute. And that triggers a massive internal distress. And these animals would develop a coping mechanism, which is an adjunctive response. They would start drinking water in that context in response to this schedule-induced delivery of food. And that drinking water response is anxiolytic. But the fact is, not all the rats, if you look at the volume of water drunk over sessions, here it would be over 21 sessions, some rats are pretty good at coping with that distress by expressing the adjunctive response towards water. So these are the green rats here. But some rats are not capable of developing the coping response with water. And what Lucia showed is that the rats that develop the coping response with water, developing that response decreases substantially their anxiety. So yes, they could cope with the distress produced by the procedure. And the rats that could not develop the coping response showed an exacerbation of anxiety as compared to baseline. Of course, they could not cope. And then Lucia introduced alcohol instead of water. The rats that had developed the coping response with water, of course, they would continue drinking, and now it's alcohol, but they had already developed the coping response with water. Interestingly, the rats that could not develop the coping response with water quickly acquired the coping response with alcohol. They relied on alcohol to learn this adjunctive response. And not surprisingly so, once they've developed the adjunctive response, they showed a decrease in anxiety as compared to baseline. And the animals that had developed the coping response show no changes in anxiety because the coping response was already developed. They did not need alcohol to cope with distress. We then challenged these animals with quinine, and we asked whether they would be willing to persist in drinking alcohol despite quinine. I would like you to bear in mind that here the two groups have had the exact same exposure to alcohol. So they've had the exact same intoxication levels every day. But when we challenged them with quinine, while the animals that had developed the coping response with water decreased by 80% their alcohol intake, those animals that had relied on alcohol to develop the coping response resisted substantially to adulteration with quinine. And that is evidence that the acquisition of alcohol use as a coping mechanism promoted a vulnerability to developing compulsive alcohol drinking. At the neural systems level, if you remember that boredom is related to dysphoria and the stress system, well, Lucia tested the hypothesis that perhaps the brain mechanism related to dysphoria, the kappa dynorphin opiate system, could be responsible for mediating this coping mechanism induced compulsive alcohol drinking. And she indeed demonstrated that if you were to challenge these rats with a long-lasting antagonist of the kappa opiate receptors, rats that had developed compulsive alcohol drinking decreased their alcohol intake, whereas rats that actually had developed adjunctive behavior with water were not influenced in their alcohol intake by the same treatment. So clearly here, it demonstrates that the endogenous system that mediates dysphoria, or one of the systems that mediates dysphoria, the kappa system, is contributing to the compulsive drinking of alcohol when animals have engaged in alcohol drinking as a self-medication strategy. So what have we seen today? That situations that promote boredom, lack of exteroceptive sensory stimulation, or social isolation facilitate the use of drugs as a coping self-medication strategy. We've seen that experiential factors in a drug-taking context, use of drugs as a coping strategy due to the perceived adversity of the context, or in a state of lack of exteroceptive stimulation, exacerbate the vulnerability to develop addiction. We've seen that the development of alcohol addiction depends on specific alteration of the GABA system in the central amygdala, and the development of compulsive alcohol drinking under CYP in a self-medication context is dependent on the recruitment of the kappa opiate system. So together, these data suggest that there are specific neural mechanisms recruited when drugs are used under negative reinforcement, the context of use of drugs in a lockdown situation, and perhaps a personalized medicine strategy would be a combination of a drug that targets the GABA system, such as baclofen, and a drug that targets the kappa opiate receptors, such as Nobianai-like drugs. I would like to thank the people who've done these extremely difficult experiments. Maxime Frissac, here, Lucia Marty-Pratt, Michael Plot, Éric Ducret, Aude Belin-Rossan, Nathalie Van Hill, Jean-Luc Cueto, with whom I initiated this work, and I would like to thank my mentor, Barry Everett, and thank you for your attention.
And thank you, David, for that wonderful talk. And thank you to all our speakers who have joined us over the last term and given us a real insight into the impact of COVID-19. We hope that you join us next term when we will be continuing our Cambridge Neuroscience interdisciplinary seminar series. See you then.